My previous lectures were based on cell biology, where I have explained you the structure of cell organelles like mitochondria, chloroplasts, ribosomes, endoplasmic reticulum. Now, eukaryotic cells, they possess a type of supportive framework inside the cell, just as most of the plants or most of the animals have their supportive framework. Chordates or vertebrates, they have supportive framework in the form of skeleton. Similarly, a cell, not prokaryotic cells, but eukaryotic cells. But there can be exceptions, like if you take amoeba. Amoeba lacks these cytoskeletal structures. And most of the eukaryotic cells, plants, animals, fungal cells, they have a supportive framework inside the cell which provides a support, which gives a perfect shape, which helps in other activities, and that is called cytoskeletal structure. So, cytoskeletal structures are the components of eukaryotic cells, except amoeba, plant cells, animal cells, fungal cells. These are supportive framework. These provide shape to the cell. These provide strength to the cell. These can help in some other functions. And one of such structure is called microtubule. Microtubules are present in plant cells, animal cells, and fungal cells. Whether it be a plant cell, whether it be an animal cell, or whether it be a fungal cell, microtubules are present. And there was Roberts was a scientist, Robert, along with an associate called Frankis. They have the credit that they discovered microtubules first. Actually, at that time they called them neurotubules because they saw them in some neurons. But later these were called microtubules. Now, if we take a microtubule, let this be a cell, cell, these are present in the cytoplasm, this is nucleus, and cytoplasm, these are present in the cytoplasm. These can be numerous, and when you will see them under electron microscope, these appear spherical in shape, just as this. Small balls like. There can be many and many in the cell, small balls. And if you will see, their outer covering can be about 50 angstroms in thickness. Their total diameter, if we will see, it can be about 250 angstroms. And this core is blank. This core, it can be about 150 angstroms. Once again, I will explain. The total diameter of a microtubule can be 250 angstroms, out of which this core, which is blank means empty, it is about 150 angstroms, and the wall which surrounds the core, membrane which surrounds the core, it is 50 angstroms. Now let us go deep under electron microscope. We will see actually this is not continuous. The core, this membrane is not continuous structure. Just I have drawn. Actually, it is formed of small filament-like structures. This type, which occur in the form of spiral. Just as this. Small rod-like structures which are called protofilaments. This type. These are protofilaments. And you have to draw 13 such. I have drawn 1, 2, 3, 4, 5, 6, 7. But we have to draw 13. such these rod-like structures, filament-like structures, these are called protofilaments. So, if we will see that the membrane of the microtubule is actually formed of 13 protofilaments which have been arranged in the form of ring. That means you will take 13 small matchsticks. This is one protofilament, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 and 13. You will arrange them in the form of ring, and this is a microtubule. And let these matchsticks will correspond to a protofilament. Here I have drawn few, and these 13 protofilaments, they are arranged in the form of ring, in the form of circle, leaving the core blank. This core is blank, and it is called microtubule. Now, how protofilaments are formed? Now, here is a hierarchy. This is microtubule, it is formed of protofilaments. Now, how protofilaments are formed? Protofilament is not actually one. These are spiral-shaped structures formed of many. In our cell, there are some proteins called tubulin proteins. Tubulin protein. Tubulin proteins bear charge. They bear charge. Some tubulin proteins bear positive charge and some bear negative. Is it okay? Some tubulin proteins bear positive charge, some bear negative. Let this be a tubulin protein, it is positive charge. Definitely it will attract a tubulin which is negative. And they form an association, this is called a dimer. Or you will call heterodimer. You can call it tubulin dimer. You will call it heterodimer. One positive, one negative. Now, there can be now association. One more will come here, which will be positive. One more will come here, which will be negative. And they form a type of rod by their association, by their combination. And that rod-like structure is called a protofilament. So protofilament is formed of tubulin dimers, positive, negative, and a type of rod is formed which is called heterodimer or tubulin dimer. So we start from tubulin dimers. First we have a positive tubulin dimer protein and negative tubulin protein. These associate, these combine due to their charge. They form protofilament. 13 protofilaments arrange themselves in the form of this ring, which is called microtubule. Now we need some type of material like GDP is there, guanosine diphosphate, GTP, guanosine triphosphate. These two are very important for their assembly. These help in assembling of dimers, these help in formation of protofilaments, then these help in the association of the protofilaments to form a microtubule. Sometimes you know that microtubules, one of the important function of microtubule is cell division, mitosis and meiosis. This is an important cell function. Microtubules are very much essential for cell division, mitosis, meiosis. And what these those dealing with agriculture, plants, etc., what they do to increase the number of chromosomes? They use a chemical called colchicine. Colchicine is a chemical. This chemical is sprayed when there is division of the zygote, mitosis of the zygote. They spray it, and that prevents this assembly. It does not allow the formation of microtubule because this system doesn't work. And when there is no formation of these microtubules, there will be no mitosis, and mitosis fails, and hence they double the chromosomes, which you call polyploidy, and which is very much essential to produce new breeds of the crops. So this was one of the cytoskeletal structures of the cell, which is called microtubule. So microtubules are actually formed of protofilaments. 13 protofilaments. It is 30. 13 protofilaments are arranged in the form of ring, which form a microtubule, and a spherical structure is formed with diameter of 250 angstroms. A core is there, black core 150 angstroms, and the membrane gets a thickness of 50 angstroms. The protofilaments itself are formed of tubulin proteins. We have these tubulin proteins in the cytoplasm, which occur in the form of charged proteins, positive, negative, and they can assemble to form heterodimer or tubulin dimers. Then these tubulin dimers, they assemble and they form protofilaments. So the actual process starts from tubulin proteins. So tubulin proteins are the basis of microtubule. Actually, microtubule is a structure, but the base is tubulin protein. Just as we are someone is going to construct a house, bricks are the basis. Bricks form the base. Similarly, these act as bricks, and these help in the form of microtubule. And the most important property of the microtubule is assembling and disassembling. Assembling and deassembling. That means these can assemble to form a big structure, and when they are not needed, they can disintegrate, they can deassemble. And the GTP system is important. Guanosine diphosphate, guanosine triphosphate. It is important for assembling and deassembling. So this was today's topic, cytoskeletal structures of cell microtubule, and in our next topic we will see one more cytoskeletal structure, which is centriole, and we will see there how centrioles are formed. And actually microtubules are important for the formation of centrioles. Microtubules are important for the formation of cilia and flagella. So all is these act as cytoskeletal structures, but the basis is tubulin proteins. Thanks.